start is always sacred. The start is where it all begins. And CSIR NET preparation always begins with Unit 1. Yes, Unit 1 is one such syllabus, a part of the syllabus where we all get stuck. We start with it with full energy and then we get stuck. Tell me, is that not the truth? We think that CSIR NET Life Science Unit 1 was made as a barrier to the rest of the 12 units. Isn't it right? Sometimes it feels tough, sometimes it feels confusing, and sometimes we feel, why the hell they have this particular part of the syllabus? But let me tell you a very important story here. Unit 1 is that part of CSIR NET Life Sciences syllabus which connects you from physics to chemistry to biology. This is that part of the syllabus which keeps you rooted into the grounds of science. It's deep rooted within all of us. So if I have to tell you that you have to revise CSIR NET Unit 1 today, or you have to start, and like I said, start is always sacred, you will need a lot of hand-holding, you'll need a lot of tips, a lot of techniques to master it. And that's what we're going to do in this fresh series where I'm going to come from Unit 1 to 13. Okay, I'll try all of them before the exam happens. And I'm going to include with every video a free sample paper download. Okay, so the link for that will be given in the description. Okay, but before you go and jump to the sample paper download, please watch this video because I'm going to share a lot of insights on how to accelerate your revision process for CSIR NET Life Sciences Unit 1. So Unit 1 is all about biomolecules, right? It has got a tadka. So don't skip it. Here's the caution which I want to give. You cannot skip it. For those who think, okay, let me skip it. It is interrelated, interconnected to almost every unit of CSIR NET. And yes, it has got a tadka of chemistry, right? It is that part of your syllabus which if you decide to skip up, you are doing a grave mistake. Don't do that. Never ever skip Unit 1. That's the first tip which I can give you today. Don't skip this. Okay, fine. I'll not skip it. But how do I accelerate it? What are the things I should study and revise? Let's talk about that. So here is a part of the syllabus which you must revise from Unit 1. Okay, so you have the stabilizing bonds between macromolecules such as proteins and DNA, which is van der Waals forces, hydrogen bonds, and hydrophobic interactions. A lot of question comes in from this part of the syllabus. You should have a good grip on this. Moving ahead, amino acid structures and behavior according to different R groups, properties such as chirality, terms such as isoelectric point, pK values, and various equations related to this. Okay, so you must not skip this part. Okay. Fine, there are more, but as of now, yes. So stabilizing bonds between the macromolecules, which is van der Waals, hydrogen bonds, hydrophobic interactions, okay. Don't skip. Amino acid structures, very, very important. Remembering the structures is also very, very important. Behavior according to different R groups, okay. What is the chemical behavior of these amino acids? Properties such as chirality, okay. The mirror images and isoelectric point, pK values, very important. Next part of the Unit 1 which you should not skip is protein conformations, okay? Dihedral bonds and Ramachandran plot. By heart Ramachandran plot. You must know everything about it. And almost every part C question, just like, we have to sniff it out. Wherever there is a part C question or part B question related to Ramachandran plot, pick it up, solve as many as possible, okay? Determine different conformations and their sequencing. All this you must remember by heart. Now, protein folding, thermodynamics, denaturation, biological functions, enzyme kinetics, especially the equation, Michaelis-Menten equation, knowing the Vmax, Km, very important. It's a must to study along with inhibition processes, the graphical representations and formulas. Again, now this is where students get confused, okay? The moment there is a graph, we either feel like skipping it or just skimming through it and marking a wrong answer. Never ever take graphical questions lightly. Data is going to be a part of your research world later on. So that is why CSIR is obsessed with data-driven questions and applicatory questions, because that's how you're going to do research. That's how they will maintain the quality of research. And that is why this barrier called CSIR NET is there. So yes, you have this part, which is again very, very important. Moving ahead, important parts, metabolic pathways of carbohydrates, including glycolytic and TCA cycle, right? TCA cycle, by heart this, okay? Practice as many times as you can, all the steps, all the catalysts, all the regulations, all the, feedback animations you should know, okay? Enzyme cofactors involved, their stoichiometry, etc., and ATP synthesis throughout, along with the inhibitors which are used at ATP synthesis. It's very, very important. So please note this down. You must revise. But how to revise, all that I'm going to tell you shortly. Next, numerical questions on molarity. Bahut mushkil. It comes all the time and people get confused. pH, buffer, thermodynamics, delta G calculation, various cellular processes. All this, again, data-driven numerical questions. छोड़ नहीं सकते आप. You can't leave it. You can't skip it. You can't skim it. You have to study. You must revise all this in detail. Now, ठीक है, revise कर लिया. अभी और क्या? What else to revise? मैं पूरा syllabus नहीं दे रहा हूँ. I'm just picking out some important aspects here. Okay. So you have the carbohydrate structure again. The concentration of epimers, anomers, glycosidic bond formation, type of bonds between di and polysaccharides, glycosaminoglycan examples and functions, all this you have to know. Lipid structure with special emphasis on membrane lipids and adaptation to temperature, because under temperature the topology changes. Now, nucleic acid topology, there is twist, rith, linking number and structure and nucleotide biosynthesis. All this you must know. You must know all this by heart. I'll just move over to the screen so that probably you can note it down or take a screenshot. Okay, so this is all about what to revise. This is all about, okay, these are the facts which we have to do, but how to do, let's talk about that. Now, first thing, get a good grasp on basic chemistry. Without chemistry, you are nothing, okay? Sounds a longer way, but actually this helps to understand and accelerate your concepts of biochemistry. Biochemistry ko aap easily samajh sakte ho if you know chemistry properly, okay? So how exactly chemistry behaves, which will help you understand biochemistry in an easier way, in a better way. But how to learn chemistry? Logically, very important here. Logic is very important, okay? Next, better you will understand, the faster you can revise. So that's the next step. You have to know chemistry first. That will help you accelerate your biochemistry understanding, and the better you will understand, the faster you can revise. So at the first time only, if you're studying, spend as much time as possible, okay? Now, concepts such as atoms, molecules, bonds, functional groups, oxidation, reduction, nucleophiles, stereochemistry, all this is a must. Now, these concepts you will see throughout the syllabus, okay? Anywhere they may combine stuff. They love combining concepts. They love combining stuff. So, and especially people getting confused in oxidation and oxidation reduction numbers, and then stereochemistry. Again, another confusing aspect. Don't get confused. Practice as much as possible, okay? Now, another thing. Make a list of important topics and subtopics, which I just showed you. Make a list and try looking at the subtopic and recollect the points about it. Okay. So the moment I say, okay, oxidation reduction, तो जो भी याद आता है उसके बारे में try to recollect. Okay. So just write the names of the concepts. Okay. Just by looking at the concepts, you can do that. In fact, I can show you an example of that we have built at Biotecnika. We call it the concept cloud. Now, what is that? Let me just pull that out for you. So here, if you see, okay, so this is probably, so let me see if it is visible here. Yeah, it is visible here, as you can see. So the moment you see chirality, you can, figure out, okay, what exactly it is, or whatever salvage pathway or polysaccharide. So, the moment you look at it, try to recall. That's the first thing which I can share with you today. The next is you can write down all the points as well. So while recollecting, write down. Remember, when you combine 3 sense organs together during revising, you revise better. Okay, the recollection happens faster. So what exactly are the 3 sense organs? The eyes, okay, ear and hand. So use your hand to write, ear to speak. So speak while you are writing, and eyes keep seeing. So when you are doing all the 3, the chances of remembering will be faster and better, and you'll retain longer. Now check with this textbook or Biotecnika's PPTs if you have the access to the study portal of Biotecnika. Okay, so you can go ahead and check that out. So there you will find all the points which you might have missed, or it might be you got any point wrong. So when you go into the PowerPoints which Biotecnika has given in the study portal, they all are given in the bulleted format, just like short notes. So while looking at that, your revision happens. So that's another tip I can give you. Now go through the topic once again. That's again another shortcut I can tell you, is just skim through, open the textbook, skim through and look at the bold headlines and see, okay, iske bare mein mujhe yaad hai ki nahi. I just try to recall. Okay, now this will help you in a systematic and all-inclusive revision approach. Most of the time what we do is we revise by solving questions, but that's the wrong approach. Looking at the bold words in the textbook, okay, that acts like a concept cloud, which I just showed you. So that way you can do it. Now next is your flowcharts. Now what exactly is a flowcharts? Make simplified flowcharts. Now pathways or cycles. Now what happens here? If you make this, you will be in a better position. Now if you have not made it, you will know that making simplified forms of metabolic pathways will help you revise faster. And that is where, if you already have the access of Konceptika in your study portal, it will help you. Now what exactly is Konceptika? Let me show. So Konceptika is a book where you have flowcharts pre-made. For example, this is a flowchart. I can just show you pH and buffer, or this mitochondrial ETC, which is electron transport chain. So just by looking at it, you can remember. And this is a colored book, and the e-copy of this is given in a study portal, which you can revise through. Okay. Now, remember, although it helps to have all the information in one page, revising becomes complicated with so much information. So what you should do, you should keep it simple. Okay, reactants, products and enzymes. If it is too much of text in your flowcharts, you're anyways going to lose your concepts. You'll not be comfortable revising from it. So keep it simple, and later on check the other details from textbook. So quick revision through flowcharts, and then again re-revise through the books. That's a sandwich model you can follow. The next, take colored printouts of processes, structures, diagrams, and pathways. Now, nowadays you have almost every standard books, PDF or something available on internet. Probably you could take a printout, or just take a snapshot from the mobile and take a printout of that and keep it in front of you in the in your wherever you study. So what happens here? Learning and revising from colored diagrams is both fun and easy, and that's where your pictorial memory kicks in. It makes your recollection faster. So let's take example. If you are revising a protein folding process, so what happens? If there is a colored diagram, it is easy to perceive, and important parts can be highlighted as well, right? So this is one of the best ways. Now this way you don't have to put extra effort to search for the details, right? So that's where the colored printouts will help you a lot. Moving ahead, now revise from flashcards. Now what are flashcards? They're just cards with one word written at the back. It has got stuff. So if you have not made it, you can always take it from Biotecnika. Otherwise, the virtual flashcards are also there in Biotecnika. So what it does is, it is very easy to carry. It's very easy to revise quickly, and one concept at a time it is, right? Basically, that's how we should study concept in a systematic way. Now, write the topic name or the word on the one side. On the other side, you can write the description. In fact, that is why in the concept cloud charts, we have given at the back a blank space. Okay. So if you see here, let me just bring this up again in front of you. So this side you have the words, okay? And the other side it's blank, so you can write your short notes here. So that's the benefit of these flashcards or concept cloud cards, okay? Now write the topic on one side, the description on the other side. 4 cards can be made out of one A4 sheet. So it's as simple as that, and you can just shuffle or juggle. Now restrict to 3 to 4 short points per topic. Don't write all Hanuman Chalisa in that. That's not going to help. Concise and crisp notes behind every word. Now you can also draw molecular structures on the cards, one amino acid, one nucleotide structure, or one card at a time. So this is how you do it. But suppose you have less time. In that case, you can always go for Biotecnika's virtual flashcards or hard copy flashcards. But this is one of the best ways you can do. Now, focus on properties, functional groups of structures instead of memorizing. Now, what happens with the functional groups? The moment you know the functional group, you can easily say what will be the behavior of this molecule, okay? What will be the property of this molecule, right? So instead of memorizing, you can just look at the functional group and you can say, okay, what exactly it is. So no need to draw and redraw structures to learn or revise them. Try knowing the important functional groups like NH3 or CH3, something like a CH4. So what happens is if you know that, then you can easily know their properties, their behavior, and their contribution to the function, the behavior of that molecule altogether can be deduced from that functional group. And that's one of the best ways to remember. Next point which I would like to make here is revise from comparison tables, okay? So like I've always said, draw a table, say you want to distinguish between DNA and RNA, okay? So RNA and DNA, right? So what's the difference? Difference 1, difference 2, right? So like that you can write it down. So that's the benefit. So revising from a comparison table and recollection is easy because one side point you forgot, the other side you can always remember. So this brings me to another product which we have created, another book which we have created, and that will help you in case you have less time or you don't have time to make it. So you can always refer to concept tables like this. So this is a very handy book. You can easily carry. Okay, it's hardly the size of my palm. So this is a book which you can carry. This is called as concept table, okay? And it has got concepts like this, if you can see here, okay? So for example, glycogenolysis and glycogenesis, or Krebs cycle and glycolysis. So you can just look at one side of the pointers and you can always correlate and remember the other side, okay? So that's one of the best ways to recollect and revise for Unit 1. So, for example, if anyone compares the process of glycolysis and gluconeogenesis or a competitive and uncompetitive enzyme inhibition, it will help you revise the differences and similarities both faster and better because correlation helps. Okay, एक साइड भूल गए दूसरा साइड याद है ठीक है वो भी याद आ जाएगा. That's how you can, in fact, you know how I used to remember is, okay, there were 8 points, why I'm remembering only 7. That's how you can correlate and remember. So that's where it is. Now another very important point which I would like to bring to your attention is pay special attention to thermodynamics. बहुत ही dangerous है. Thermodynamics, regulation of inhibitors of a particular process. Now if you're studying, suppose, nucleotide metabolism, so it is important to revise the important points such as the regulatory step, okay, how exactly it is getting regulated, the rate limiting step and what is causing that rate limitation, inhibitors of the pathway, and spontaneity of the steps, how fast it's happening, what is the rate of reaction, all that you should know. Now this applies for all the metabolic pathways, not just 1 or 2. So you should know all this for all the metabolic pathway, and this is one thing which I want to highlight here. Next, data, right? Your life will be full of data. The moment you qualify as a PhD researcher and you start researching, your life will be full of these kind of graphs. So knowing the basic numerical speed maths, molarity, normality, unit conversion, etc., etc., is very important. Otherwise, you will think it is gram, but it will be kilogram. Okay, you will think it is meter, it will be kilometer. It will be CGS unit, you will think it is MKS unit. That's the kind of danger you can be in if you don't pay attention here. Be familiar with the type of graphs which is there, how to analyze and interpret them, whether it is a pie chart, whether it is a bar graph. Revise the graphs from standard reference books. Very important. Now, one thing what I have seen about these kind of graphs is CSIR loves to play around with graphs. In fact, almost every competitive exam loves to play around graphs. And why is that? Because research life will be full of data, full of graphs. So they want to know, they want to make sure that the only students who are very, very strong in data interpretation get in. And that is why they're going to confuse you. And it's very, very easy actually to confuse you with, data related questions. Okay. So whenever you see data related question, just jump and solve it. Okay. Before the exam, okay, and so that during the exam when you see it, you will not be scared. Okay, that's very, very important here. Now practice previous year question papers. You can get an idea about what I'm talking and how important this situation is. The next thing is flowchart and classification tables. Okay, so classification tables or charts are very important. Now, whether you're studying classification of carbohydrates or lipids or types of cofactors, protein function, it is recommended to make a consolidated chart or a table. Okay, if you do that, if you just create a table with all that, okay, just take out a sheet of paper, use some colored pencils, draw it, it will save at least 10 minutes of your time during revision, and later on you will be in a better position to revise faster. Okay, you will be able to recollect faster because the tables are nothing but a kind of a picture, right? So your pictorial memory kicks in and you can revise faster. So again, I'm highlighting here, not just flowcharts, classification tables and charts you can create. Now, many students will ask me, what are the standard reference book which you should go for? Now, again, I'm telling you, padho kahin se bhi, but most important thing, standard books refer karo. Okay, after you have referred all of them, then only switch to any regular competitive exam books, because this is your base. What are you seeing on the screen? Take a screenshot. This is your base. What it is? The Bible of biochemistry, Leninger. Nobody can replace it. You have Voigt and Voigt, biochemistry. You have Stryer, biochemistry, and enzymes, biochemistry, biotechnology by Trevor Palmer. So these are the 4 books. Even if you have 2 of them or 3 of them, that's sufficient. 2 of them is also sufficient, but Leninger is a must. Okay, Leninger is a must. If you don't have it, you can buy from the market, buy a second hand, get a e-book, whatever it is, but you must have these standard reference book. You must study from this. And after that, you can refer Biotecnika's study material. So Biotecnika's study material, many students ask, okay, Biotecnika's printed study material, how effective it is. It will be of no use to students who don't know ABCD. These books will teach you the ABCD of Unit 1. After that, if you jump to our study material, it will be super easy to revise, because that is where we have implemented all the techniques which I just told you. Okay, but what if you don't have time for all that? You really want to jump into revision? I have a super solution for you, a super computer solution, which will accelerate your preparation very, very fast. And that is right here. We call it Konceptika flowcharts. Okay, Konceptika flowchart is another way of telling you that you have less time and you have a lot to accomplish. So all you have to do is pick out this book. Pick out this book, hard copy or soft copy, and you can study. Now, don't buy it if you are getting attracted to it. You should buy it if you feel that this will be useful to you. Okay, this book is only for those who think this book will be useful for them. Just don't buy the book and keep it on your side and think that, okay, you'll qualify. That doesn't happen. These books are colored, okay, and these books are very highly effective, efficient, and colored and created in a very, very scientific way, so that you can just look at it and you'll remember the concepts. But it doesn't do the magic unless you open the book and study. Okay, so this is Konceptika flowchart book, Konceptika Lite. Moving forward, we have the Koncept Cloud, which I have already showed you. Let me show that to you again. This is the Koncept Cloud. You can pick out anything from the cloud, any words, and you can just remember this. This is the entire bundle for I think Unit 1. So you can just look at it and you can study. No, this is not only Unit 1, this is the entire thing. So you can just look at it and you can study. So suppose unit 7 you are studying. So, okay, by just having one look, you can say, okay, sickle cell disease, myocarditis, pacemaker cells, pericarditis, desmosomes. So you just look at the bigger the word, the more important it is. So you should be able to recall faster, right? So that's about concept cloud. Next, we have the concept table. Again, I have showed you that as an example during the tips and tricks. This is also a very good way to prepare, which is just by comparison. Okay, so you have the comparisons here, you can see here, right? So you just pick out any concept you can compare. For example, commitment, determination and differentiation from unit 5, or you just take out dichots and monochots, or let's pick from the unit 1 which we have talked today, Krebs cycle, glycolysis, or glycogenesis and glycogenolysis, or kinases and phosphatases, catabolism and anabolism. You can just compare and remember. So that's about concept table. And then comes the concept wheel, which is all about your central idea and its features. So generally, there are things which you can compare, and then there are things which you cannot compare, right? So for all those things which you cannot compare, but they also are important, for example, amino acids, right? So let's pick this amino acids. So so in the center it's written amino acids, and other sides, what is the other features of this which you should remember and recall. So this is where the concept wheel comes. Again, this is a very small book, a size of my palm, and you can easily revise. The entire syllabus is right inside these books. Okay, now please don't buy it because you're attracted to it. Buy if you feel these will be useful in your preparation, because at the end of the day, please don't waste time and money on stuff which you're just attracted to. Get going and buy books which you think will be useful. And in my experience, these 4 have been really, really useful to all our students, whoever has accessed it. So having said that, now is the time to conclude this video and tell you that whatever you do, okay, don't skip. Don't skip unit 1. Revise smarter, create memory milestones like I showed. You can yourself create it, or you can buy whatever you do, but your aim to achieve success cannot happen unless, let me repeat, unless you take unit 1 seriously. You have to revise smarter, you have to study in a better way, and that's where success will be. Because if unit 1 is clear, the biggest barrier is clear. Unit 1 and unit 13, they're the biggest barriers you have, apart from ecology and evolution, right? Unit 6, 7, those are easy ones. And if you are going into the exam without 7 to 9 chapters and 7 to 9 units covered, you are in a danger zone, because this is a competition. There's no pass marks, this is cut off, right? So don't take risk with your unit 1. Prepare with the tips and tricks which I shared with you. And now's the time for the free download for the question paper. So the link is given in the description. Please go ahead and download it, and please join our Telegram group, because that is where we share a lot of question papers on a daily basis. Okay, and right now the exam season is on. Almost on a daily basis we are sharing a lot of PDFs for preparation. So go ahead, join our Telegram group. The link is given in the description again. Thank you so much for watching this video, and have a good revision time. Take care. Bye bye.